Well, good morning, everyone, and welcome to this NERA webinar. Today, we are delighted to share with you the key results and findings from a two-year collaborative research project, which aim to improve our understanding of the radiological impacts of mining on flora and fauna in Australia's arid environments. Before we start, I'd like to acknowledge the traditional custodians of the land which we are all meeting on today, and also the traditional custodians of the lands on which this project was conducted, and pay our respects to their elders past, present, and emerging. By now, I'm sure you're all familiar with functionality of Zoom for webinars, but just a quick refresh. There are a number of buttons at the bottom of the screen. The Q&A function, if you have any questions for the presenters, then please post them there. All the speakers will be on a panel session at the end, and we aim to get through as many questions as possible during the available time. If you see a question you are interested in, please upvote it. We will endeavour to address those with the most votes first. And if you have any technical issues, please use the chat function to let us know, and we'll address them in the background. Okay, just before I introduce the first presenter today, I would like to provide you with a quick background to NERA and why we are supporting this project. NERA is the Industry Growth Centre for the energy resources sector. We were established by the federal government in 2016 to take a strategic and industry-led approach to driving innovation, productivity, and competitiveness across the energy resources sector. Like the other 5 growth centres, we are a not-for-profit organisation governed by a board of industry leaders. As is evident by the collaboration in this project, we work across the entire sector, including industry, universities, governments, and innovative small businesses, to support the cultural change and new thinking required to overcome barriers to collaboration and innovation, and to assure that Australia's future energy resources sector is globally competitive, sustainable, and diverse. So why is NERA supporting this project? Well, I think there are 3 main reasons. Number 1, it's a strong collaboration with a good mix of collaborative partners. We have Flinders University as the primary researcher, a strong mix of operators in BHP, Heathgate Resources, and Oz Minerals, who are all willing to share information and data. And we have government organisations in our PANZA and ANSO to help guide the project and assist with analysis of samples. And finally, we have JRHC Enterprises, a small business but an extremely well-respected radiation management consultancy, who are able to provide an overarching industry perspective throughout the project. The second reason is that it addresses a clear industry problem. Currently, radiological impact models for non-human species rely heavily on Northern Hemisphere datasets, which creates some confusion in communicating the potential impact of relevant Australian proposals. The results of this joint industry research national authority project will contribute to the national database of radiation concentration ratios of flora, fauna, and soils in areas of Australia not previously surveyed. The database will be an invaluable resource for industries in the arid and semi-arid regions of Australia that are required to assess their environmental radiological impacts as part of their whole of life activities, including approvals and relinquishment. And thirdly, the project has also built radiation management skills and knowledge for the future workforce in the mining industry by supporting several new research opportunities for honours, masters, and postgraduate positions. Now, our first presenter today is Jim Hondros, who many of you would be familiar with. Jim has over 35 years experience in the resources industry in a range of roles, including operations, corporate, and consulting. He has worked in radiation protection, occupational hygiene, safety, and environmental management. More recent, Jim has been working with the International Atomic Energy Agency, conducting reviews of guidance documents and training in various countries. Jim also regularly undertakes voluntary work for the Environmental Defenders Office and provides advice to various Indigenous groups. Jim is currently Vice President of the Australasian Radiation Protection Society and is co-chair of the International Radiation Protection Association Task Group for Norm in Industry. Today, Jim is going to provide us with an overview of how the resources industry assess radiological impacts to the environment and how the results of this project will benefit industry moving forward. Over to you, Jim.
Thanks, Tim, and good morning to everyone. Rose and I have been involved in this project since its commencement a couple of years ago. Our background, as Tim mentioned, is that we have conducted environmental, radiological risk and impact assessments for many resource companies, both here in Australia and overseas. And I'd like to give a short presentation from the perspective of an industry practitioner, and also discuss why this type of work is important for the resources industry. Let me start by saying that we do not represent any one company or the industry as a whole. However, we have wide experience across industry, both here and overseas, and are able to make some general comments from the perspective of industry. The first thing is the resource industry is really diverse. It's very diverse, and naturally occurring radioactive material occurs everywhere and is present in all mined material to some extent. The act of processing that material will tend to either concentrate radionuclides in products or in waste streams. When the concentrations of radionuclides exceed a certain level, then the material may be legally classified as radioactive and therefore trigger an assessment requirement. We're all familiar with the uranium mining and mineral sands industry sectors being classified as radioactive. However, elevated concentrations of radionuclides also appear in many different other resources, from gold to bauxite, from copper and tin to rare earths. Elevated concentrations can also occur in the oil and gas industries. Understanding the impacts provides confidence for the company, provides confidence for regulators, and more importantly, provides confidence for the communities. Once a material is classified as radioactive, then there is a requirement to assess the risk. The international body responsible for setting radiological standards is the International Commission on Radiological Protection. Over recent years, the approach to environmental protection has changed. Previously, it was accepted that if people were protected, then the environment would be protected. However, there is now a recommendation by the ICRP to demonstrate that the environment is being protected. It's also important to note that in radiation protection, we aim to protect individual people. However, in regards to the environment, we aim to protect species. So in practice, when radioactive material is present, there is a requirement to demonstrate that plants and animals are protected. For new operations, the impacts can be modelled or predicted. For existing operations, the impacts can be measured. When conducting an assessment, it is important to consider local conditions and local species. The most important factor in any assessment is the concentration ratio, which is a measure of the ratio of the concentration of an element or a radionuclide in a particular species compared to the media. For example, the concentration of a radionuclide in a kangaroo compared to the concentration of that radionuclide in the soil or water. It's also important to ensure that a graded approach is used. This means that if the impacts are low, then a shortened assessment is undertaken. This is consistent with the idea of ensuring that controls are commensurate with risk. Assessments themselves are really not that difficult, and there are a number of tools available, and I'll show these next. So, how do we do an assessment? Basically, we determine the change in the media concentration, that is the soil or the water, due to an operation. For example, dust fallout or water releases. We then take into account the concentration ratio for the particular species in that media. If we don't have species-specific concentration ratios, then there are international default values that can be used. Also, if the default species are not present in the area we are studying, then we need to align the local species to the default species. Already you can see there's quite a few assumptions in this. Then, using internationally recognized assessment tools, for example, ERICA or RESRAD, the assessment is conducted. The results of the assessment are compared to predefined criteria. If the results exceed the criteria, then a higher order assessment is conducted or controls are implemented. As you can see, there is a reliance on international default values in the absence of local data. This is important because it assumes that international conditions and concentration ratios are appropriate for the Australian conditions. As mentioned, a few pictures here, we can model changes in media concentrations using air quality modelling. On the left is the output of some air quality modelling for a proposed mine. You can see the contour plot which shows the levels of dust deposition. And on the right, we can measure changes in media concentrations once a project starts. And this picture shows a dust deposition gauge. When it comes to concentration ratios, we have default values, as I mentioned, as shown in the left document through the IAEA publication 472. We also have some local Australian concentration ratios. On the right is a publication from Arpanza, authored by Gillian from a few years ago, which collated data from a number of mine sites around Australia and produced some Australian concentration ratios for different species. I would like to mention that there has been extensive work undertaken on concentration ratios in the semi-tropical Alligator Rivers region by Shay Doring and Andreas Bollhofer. Their work is specific for the semi-tropical regions of Australia. Which brings us to the topic of this research work, the determination of concentration ratios for the semi-arid region of Australia. This is relevant work for more accurately assessing radiological risks in the semi-arid region of Australia. And as I mentioned, the assumption to date is that the international default values are appropriate for Australian conditions. So what are the benefits for industry of this type of work? Well, we know that many companies are already undertaking environmental radiological impact assessments, and generally we know that the risks are low. However, there is an ongoing need to assess the risk to demonstrate that the risks are low. And again, as I mentioned at the beginning, importantly, to communicate that those risks are low. Using local factors in risk calculations improves the accuracy, relevancy, and credibility of assessments. It is also relevant to note that for many operating companies, data exists. There is fundamental monitoring data that is available that can be used to develop some of these concentration ratios. However, the overall aim is to move towards assessment using local values, which improves the way that operations are managed. So in summary, there is a requirement to demonstrate that the environment is being protected from radiological impacts. Generally, many companies are already doing these assessments. This work improves the assessments by providing local species data, which can be different from international default values. My observation is that the collaborative nature of this work has been a significant outcome. If we have better information, we are able to better progress towards a graded approach to regulating operations. And the local data improves how and what we communicate. In closing, I would like to say thank you to Rachel, ex-Flinders now, and now Melbourne Uni, who has held together the research over the last 3 years. And also thank you to Tim from NERA, who oversaw this work. Without these two, the work could not have been conducted. Thank you.
Thank you. Thanks, Jim. Thanks, Gillian. So next up is Dr. Angel Ray, a research associate in environmental radiation at Flinders University. Angel received her PhD in Sciences from the University of Adelaide in 2018 on her work to understand the global gold cycle. Angel's current role focuses on the project we are discussing today, and along with Rachel Popelka-Filkov, Angel has been a key driver to ensure the delivery of this project, and she will now provide us with an overview of the project and the key findings. Over to you, Angel.
Okay, so I'm assuming everyone is currently looking at the full screen of my presentation. So for today, I would like to share with you the results and outcomes so far on our project on improving the relevance of international radiological assessment tools in Australian arid environments. Again, the project is funded by NERA, our 3 industry partners, BHP, Heathgate, and Oz Minerals, and the radiochemical analysis was performed at ANSTO and Arpanza, and the project is supported by JRHC, Arid Recovery, and Popelka Filkov's lab at Flinders University. Okay, so why was the project conducted? We have conducted the project because we want to expand the knowledge on environmental radiation profile in order to protect wildlife diversity. Ultimately, what we want is to improve the Australian-centric capacity for environmental radiation impact assessment in terrestrial environments. In order to do this, what we have done is select three regions, the Flinders Ranges, Roxby Downs, and Pernati region as our sampling site. We have collected soil at 0 to 10 centimeter depth, flora clippings, and also opportunistic fauna samples. These were then prepared and measured in order to obtain the concentration ratio, which is the activity concentration in biota divided by the activity concentration in media. And in our study, that will be the soil in terrestrial environments. The question here is, why are we using concentration ratio? Just to reiterate what Jim Hondros mentioned earlier, we are using concentration ratios because this is a parameter that's currently used in international radiological impact models. Another reason is that concentration ratios allow us to compare data on organism uptake in different sites and also allow us to compare data across Australia to international reference data. Okay, so what we did is once we have selected the region, is we have identified 4 sites to obtain 5 soil samples in each site at 0 to 10 centimeter depth. This sampling procedure is a standard approach used in international studies. We have also obtained 3 individual plant samples per species and at least 2 plant species per site. Opportunistic fauna samples were also obtained from each site, and these were prepared and then freeze-dried at least 24 hours for alpha, gamma, and NAA. Majority of the radiochemical analysis was performed at ANSTO and Arpansa, and the details of the procedure will be published in our Flora and Fauna paper, which we anticipate to be published hopefully in 2021. So please watch out for the announcement on when this will be published. So in this study, we have performed a total of 8 sampling trips between 2018 and 2019. And this sampling procedure and the sample collection was made possible because we have worked with BHP, Heatgate, and OZ Minerals, and also with Arid Recovery. So in this photo, we have Nick and Mark from the Pernati region identifying the plants, helping me identify the plants, and also collect soil samples. We're also privileged to have a free tour with Marty to the Paralana Hot Springs to appreciate the environment and geology of the Paralana Spring in the Flinders area. Another benefit that we receive from working with a mining partner is we have a privilege of visiting their mining operation. Here in the photo is Jim, Rachel, and the students looking at the mining site, mining operation, and also Daniel, Sam, and Will at the processing plant. And again, these are just among the benefits of working with mining partners. Within this 2-year project, we have generated more than 150 flora. We have sampled more than 150 flora samples that generated 1,500 data points, 25 fauna samples, and 150 soil samples generating around 1,100 data points. And this was made possible because we have worked with this team at ANSTO with Dr. Matt Johansson and Dr. Tim Payne, and we also received additional support from Dr. Gillian Hirth and Liesel's radiochemistry team at Arpansa. And what I can say also is that everyone shared best practice approach during the method development for biota, which resulted to this huge amount of data generated in this project. The data set that was generated will be published as a supplementary information in our Flora and Fauna paper, so please wait for the announcement on when this will come out. And the Australian specific database is still under discussion with our project partners. And as soon as this is finalized, we will let you know. And also, finally, there is an opportunity here for collaboration. If you have any Australian data on radiological monitoring that was done on plants, animals, and soil, and you want to include this in the database in future publication, please get in touch with us or any panel in this webinar, and we will discuss how we will move forward with this project. We're also developing novel approaches in environmental radioecology in order to develop non-lethal ways of monitoring the wildlife. Also, please follow us on Twitter if you want to learn about research updates and also when publication will happen. Finally, I would like to thank all the organization and all the mentors who are watching me, all my mentors watching me right now. Um, Thank you for your help in making this project and helping me in completing this project. And I would also like to thank Tim Duff, the project manager at NERA, for the generous support that you have given us in this project. And also thank you everyone for listening, and I hope you enjoyed our presentation. Thanks, and back to you, Tim.
Excellent. Thank you very much, Angel. Okay, so just a reminder, if there's any questions, please put them in the Q&A function below. I see a couple coming in, but we've probably got time for a few more than that. So our next presenter is Dr. Gillian Hirth, who is the Chief Radiation Health Scientist and Deputy Chief Executive Officer of our PANZA. Gillian completed a PhD in environmental radiochemistry in 1999, which focused on the transport of the uranium decay series in the environment and their transfer to biota. Dr. Hirth is the current chair of and Australian representative to the United Nations Scientific Committee on the Effects of Atomic Radiation and the Australian representative on the International Atomic Energy Agency Commission on Safety Standards. Dr. Hirth is also a member of the International Commission on Radiation Protection Committee 4 and is a member of the Board of Council of the International Union on Radioecology. Today, Gillian is going to talk to us about what's next, where do gaps still exist in the database, and where else could we add value. Over to you, Gillian.
So thanks, Tim, and congratulations to Angel, Rachel, and all of the team in getting to this point in the project. It's been a huge effort, particularly during 2020, that has involved the support of many dedicated people who I'd like to acknowledge. And I'd also like to note it's collaborative projects like these that provide new data and insights that help give organisations like our PANZA as a regulator confidence that environmental assessments in Australia are based on quality data representative of Australian environments. This supports not only new assessments, but as mentioned, it can improve public understanding and confidence in previous assessments that have been done over the last decade. It also provides us with more information about where there might be differences between concentration ratios collected from other climatic regions or hemispheres, and enables us to understand whether these differences are significant or not. Having Australian-specific concentration ratios for arid Australia will support clear and confident decisions, as environmental assessments are more robust and can provide greater confidence to the broader community and public. It also provides an understanding of the natural background environment and supports the communication regarding the impacts to a range of stakeholders, including government and communities, and allows the comparison of radiation risks to a range of other risks. So the main outcome of this work has been to continue to build a new data set on arid region flora and fauna, and not only to increase the data numbers, but it reduces uncertainties. And it can also be compared to historical data, which, if consistency is seen with older data, it can also assist in validating and increasing confidence in that older data. So let's move on to what's next. Where are the gaps and where could value be added? So in thinking about what's next, our PANZA will continue to encourage further literature review and data sharing across norm industries. If there is data that's being collected for other purposes, it's important to maximise the use of this information. Interestingly, it's getting close to a decade since I undertook the review that Jim mentioned in his presentation, which looked at historical non-human biota CRs and consolidated information from the 1970s through to 2010. And that project was undertaken in 2012-13. So it's getting close to a decade ago. Results have also been published by researchers with the Alligator Rivers region, and now this work from Angel and the team. And all of these publications are resources that can be utilised to understand Australian CRs and how they compare internationally. For some data sets, it would be valuable to look at biota groups and nuclides and check the coverage that we already have and see if there are some categories where we have enough data. It may also be useful to see whether there's some gaps and particular areas where ongoing focus might be required. And as Angel mentioned, looking at the strategy for developing either a national database or a national periodic review process. And these are some of the things that I think we need to do and consider how this can be done in a sustainable manner going forward. So where are the gaps? I think fauna sampling in Australia is always problematic. We have unique animals, but also I think some of the strategies that Angel has identified during her work have been quite useful. And this is also where it's important to understand if we have sufficient data. It's also encouraging that there is another project already initiated to undertake a literature review and gap analysis of oil and gas norms in the aquatic environment. This is being done by Curtin University and Aberdeen University, specifically around decommissioning. And this is a national decommissioning research initiative being done in conjunction with NERA. Australia does have a significant decommissioning program of work ahead, and this is of specific interest to both ARPANZA and NOPSEMA. In relation to international guidance, the ICRP have released new guidance in 2019 on radiation protection from norm in industrial processes. And it's promoted risk-based assessments where radiation is considered in an all-hazard assessment. And whilst this has been published, ARPANZA is also considering what supporting information or guidance that might be required to support implementation. ARPANZA will also be playing an active role in the update of an IAEA safety report on radiation protection and the management of radioactive waste in the oil and gas industry. And I think starting today, the first consultancy meeting will be held a little bit behind schedule of where we'd anticipated this would be because of the pandemic, but this will be an area of work that ARPANZA will be involved in and focused on in the next 12 to 18 months. This will look at systems and arrangements, not just for workers, but will also provide an opportunity for providing additional guidance on decommissioning and the need for environmental assessment and considering that whole of life of operation and where the impacts lie, be that with humans or with the environment. And ARPANZA will be seeking industry input in regards to the development of this safety report. So finally, where could value be added? And I think the key thing is access to existing data that might already be collected by operators, but it's not necessarily published, and maximising the benefit if samples and information are available in order to be able to consolidate this into a national data set. I think compatible procedures for sample collection and analysis and guidance around, as Angel indicated in her presentation, times of year that might be more optimal, and just understanding, what was done 10 years ago 20 years ago and what's being done today, and how that might impact potential variance with data. I think, yeah, what information should be recorded. And finally, this is again supplementary guidance that could be developed as a companion to the guidance published by ARPANSA in 2015 around the radiation protection of the environment. So finally, in closing, I'd like to thank all the partners, and I'd like to thank NERA for their funding, plus also the industry partners, the university researchers, and the government collaborators. I think we should all be really proud of the work that is done in Australia. In many cases, Australia is ahead of the game with regards to the radiation environmental risk assessment approaches that we have taken, and we really set an example that should be followed internationally. So thanks everybody, and I'll hand back to you, Tim.
Thank you very much, Gillian. And I'll just ask all the presenters to turn their cameras back on so we can answer some questions that have come through. I might start with you first, Angel. You say that all the concentration data that we receive from the project is higher than the current reference data. Can you put that into sort of some context? What does it mean? Should we be worried, or?
The most important result is that the concentration ratio in our study is higher than the non-Australian wildlife transfer database. But this is the baseline. It does not mean that we are not protecting the wildlife. We need to establish the baseline data first. And in our study, we have shown that that is really unique for our ecosystem, and that this indicates that we should have this capacity to develop our own ways of having an Australian-based capacity to do the monitoring using this higher CRs as the baseline. I think Jim or Gillian can add a comment on that as well.
Okay, I think I've got my microphone on first.
You beat me.
I think this is one of the things that for the next step is really having a look at putting in context what those differences mean. You know, if we did an assessment using only Australian compared to, you know, how much does it increase us? What is the true impact? And, you know, is it within acceptable uncertainties? And so there may be, you know, I think providing that sort of guidance about, you know, if something's 3 orders of magnitude difference, yeah, it can make a big difference. If it's 1 to 1.5 magnitude of difference within the bounds of uncertainties that you might have in the CRs, how big is your data set? Is your underlying data set 1,000 samples, or is it 10 samples? And so there's all of those sorts of factors that I think can probably be articulated in the future, and hopefully not in too distant future, to really yeah, put some of those factors into the context of what it means.
I might add a comment here. I think, as you said, Angel, they're higher, but I think the more important fact is that they are different. They are different from the international levels. And I remember many years ago, almost 10 years ago, when we were doing the first set of assessments in Western Australia, and we were using the default reference plants and animals. And people would say to us, including the regulator, why aren't you assessing the kangaroos? You're assessing moose. And there was this balance about trying to get Australian relevant data. And it's taken a while, but we are now starting to see Australian relevant data. So I think, yes, Angel, it is higher, but I think they're different. The reality is they're different. And we need to know. Over.
Thank you. So we've got a question here from Blake Orr, who has asked, what are the implications for the ERICA methodology tool? And is the steady state ERICA approach appropriate? Who wants to take that one first?
I will answer that first. So the current project is not really focusing on ERICA tool alone for assessment, because ERICA is just among the many tools that we can use for radiological assessment. But I personally think that the ERICA approach will be appropriate if we use or put it in the Australian context, using Australian specific concentration ratios and Australian specific activity concentration, and not just the default values that is used in ERICA.
I think, Angel, that's exactly right. We are collecting concentration ratio data. It just happens to be able to be used in ERICA, and it can be used in a whole range of other things.
Okay, great. So Jason Christie's asked, with the animal samples, how do you know how much time the animals have spent in a local area? And did you choose species that didn't roam too far? Maybe you could expand that as well to talk about what's the ideal case, what animals would we be sampling? Angel, do you want to start with that one as well?
So with the animal samples, how much time does the animal spend on the local area? For this one, we have to consult the environmental specialist in the area, and usually they have a better idea on how much time the animals spent on the surface, or in the case of small mammals, how much time are they burrowing in the soil. Um, we prefer to choose animals that do not have a huge roaming range, but with our current study and with our current result, we have a we have a good coverage on the soil data, so that if the animal has roamed from 0 to 10 kilometers, we can still cover that with the background soil that we have obtained. So preferably the roaming range should be within a kilometer, so that we know that the soil sample that we will use to associate with that mammal will be applicable to that specific animal. But if that's not the case, we usually get the average of the whole region that we have sampled. Anyone else who wants to add?
I might just add a quick comment there that one of the constraints on the project was getting animal data, and the difficulty getting ethics approval for that. So there was some different techniques used to obtain animal data, and one of the ones we did discuss and be very frank about it was whether we could make use of situations like roadkill as a way of obtaining extra data. But the intent is not to go around collecting a whole range of animals unnecessarily, if possible.
Gillian, you might be able to add to just this supplementary question that's come through about effect studies on Australian flora and fauna. Has there been any work on that?
I would say no. There haven't been any significant radiation effect studies undertaken on Australian fauna. I think one of the things with that is you have to have biota that are exposed to, higher levels of radiation to begin with. Looking for effects when you've got animals existing in a natural environment that's not necessarily significantly elevated. It's just really about understanding what their natural radiation levels are, and yeah, it's different to doing studies in the Chernobyl exclusion zone or the Fukushima area where you have elevated levels that are measurable and comparable to a background situation.
Right. All right. Lots of questions coming through around fauna. So Jonathan Chatwell has asked, are there any tissue-specific data variations for the fauna data? And what do the error bars look like, Angel? Without giving away your publication to come out, can you just give a general answer to that one?
Okay, we have tissue-specific data for fauna, but what I have shared in the presentation are just the results that we have so far. And you're right, Tim. We will include all of the information in the publication, including the error bars in the graph. So please wait for the announcement on when this will be published. And yeah.
Great, thank you.
So Tim, can I ask a question? Are you
can.
allowed to, as a panelist, ask a question?
Yeah.
When Angel, there's been a number of questions about getting access to the report. When will it be available?
So in terms of the report, either the Flinders Uni report or the internal report, we still have to finalize the report at the end of this project, which is in December. And then we can potentially work on deciding whether we're going to create a publicly available NERA or our PANSA report. And we will discuss this with the team. But the first goal here is to publish the result first in a journal and then release the public report for anyone to view.
Yeah, and we'll make sure that everyone on this attended this webinar today is given an update when those reports are published. And hopefully by that time, we'll also be able to provide some clarity about how the data will be published, where you'll be able to access it, because it was an original intention of the project was to make sure that the data is widely available. Gillian, we just have a question around if our PANSA will be updating national guidance for industry or regulators as a result of this project.
It's certainly something that our PANSA will be considering within its work program in the future. As with all of these things, the time and the resource and finding the right people to undertake the work will always be our challenge. But certainly I think whether it's an advisory note supporting information or something more significant, we'll probably get to determine exactly what that will be, but it's certainly on our radar.
Angel, there's lots of questions around some of the variations that you may have seen. So is there much variation between deeply rooted plants or shallow? Have you seen variations in different species, the carnivores or herbivores, etc.? Are you able to expand on some of those results and findings?
In terms of the plants, we know that every plant species will have a different uptake and different uptake of both elements and radionuclides. So we will expect that that will vary. But the important thing is if you select the one that is representative of the region, then you will capture what we want in terms of the radiological monitoring. In terms of the behavior or the feeding behavior of animals, as mentioned earlier, we have difficulties in obtaining specific animals that we want to sample because we are relying on opportunistic fauna samples. So what we do with the data is just make sure that at least have an idea of what the feeding behavior of the animal is of this opportunistic fauna sample, and then correlate it to that. But again, the focus of the study is not to look at the food chain transfer, but to look at the environmental radiation profile across this semi-arid region.
Great, thank you. So there are a couple other questions that we'll take offline and make sure that we follow up throughout the rest of the week. Keen to just unpack one that came through with the registrations of the webinar though, and someone's asked if they're interested to understand sort of what the next steps are in applying concentration ratios to environmental impacts. So for example, if they're going to put through an impact assessment later early next year, would they be able to utilise this data? How could they go about that? What are your expectations, Gillian, as an assessor? Jim, do you want to start with that question?
Yep, I can sort of say as a practitioner, what I would be saying is once the data is published, I'd be using it. And there is a facility in Erica as one of the tools that you can use for making an environmental radiological impact assessment, where you can actually enter your own concentration ratios. There's also a field there to justify why you've made a change. However, I will add to that in doing that, please have a discussion with your regulator to make sure that the regulator is happy with that as well. Over.
Okay. So yes, I think you'll be using all of the available published information that's come out of studies in Australia, and identifying which values can be representative of the environmental risk assessment you might be undertaking. I think as long as you have a clearly defined overview of what your pathways are when you're doing your risk assessment, that's probably as equally as important as which CRs you're using. It's that you've defined what your exposure pathways will be, what your sources are, and then picking those appropriate concentration ratios from the available data.
Great, thank you. Okay, well, looks like the questions have dried up, and I have no intention of making the webinar longer than it needs to be. So thanks again, everyone, for joining today. And thanks to all the presenters for their hard work within the project, and also for preparing for today as well. If you do have any more questions, then please do get in contact, and we'll ensure that we answer them. Once the reports are published and the data, and we know how the data will be available, we'll make sure that we let you know. Hopefully that'll be sort of early in the new year, at least before the end of Q1, not to push anything on the other Angel. And then just as the webinar closes, there's going to be a quick survey. If you could take a few minutes just to complete that, that'd be great. And again, thank you. Enjoy the rest of your day.